The overall goals of this protocol are to cultivate and infect C57 black 6 mice with the IgG strain of Listeria monocytogenes, and to measure the bacterial load, interferon-γ responses, and survival in response to infection by this pathogen.
This protocol can help researchers screen for agents or genes that have an impact on interferon-γ responses and on animal survival after bacterial infection. The main advantage of this technique is that it is relatively simple to set up in the laboratory, even for those with little experience in cultivating bacteria. Also demonstrating the procedure will be Jennifer Ahn, a graduate student from my laboratory.
Begin by dipping a sterile pipette tip into room temperature thawed glycerol stock of the L monocytogenes IgG strain, and immediately streak the tip back and forth across a section of a brain heart infusion, or BHI plate. This is the primary streak. Turn the plate 90 degrees and drag a fresh pipette tip through the first streak, spreading the stock to the next quarter of the plate. This is the secondary streak. Then, turn and spread the secondary streak into a third quarter of the plate to make the tertiary streak. Now, turn the plate upside down at 37 degrees Celsius for an overnight incubation. Single uniform colonies should be visible in the last set of streaks between 16 and 24 hours. When the colonies appear, dispense 10 milliliters of sterile BHI broth into a sterile vented 50 milliliter tube and use a sterile pipette tip to transfer 1 L monocytogenes.